Hello, welcome to this video on cell metabolism. This will discuss the first about cell metabolism and in detail aerobic and anaerobic respiration. And also the starting point will be a few details about the importance of energy, i.e. food for our bodies, and also a little bit about diabetes because I feel it's relevant to this topic as well. So let me just share my screen real quick. And let's go on slideshow. Okay, let's begin. Cell metabolism. Okay, first of all, what is it? Metabolism describes all biochemical reactions that take place in your body, and we use this energy for anabolism and catabolism. And anabolism means constructing molecules and consuming energy. These are the processes that take small monomers such as glucose, i.e. small sugars, and amino acids for proteins, and fatty acids for lipids, and we use energy in these anabolic reactions to construct larger polymers such as lipids, carbohydrates, and proteins that we use in our cells, and this consumes energy. And the opposite to this, sorry, I forgot to mention here, is catabolism, which is breaking down large polymers such as lipids, carbohydrates, and proteins. And these processes release energy that's stored within the chemical bonds. And we use this energy to perform daily tasks such as keeping our heart beating. And when we say energy, our primary source of energy is ATP, and this is produced by cellular respiration. But the molecular fuel that we need for ATP production and cellular respiration comes from food. And the molecular fuel our body uses to make ATP is a monosaccharide glucose. Monosaccharide means simple sugar. And when we consume foods, especially carbohydrates, specialized enzymes known as amylases, they digest carbohydrates in the small intestine and breaking them down into glucose molecules. These are the monomers. And because they're small enough, the monomers are then absorbed by the small intestine's epithelial cells, and then they pass into the bloodstream directly through a network of capillaries, and they pass it to the main bloodstream and transport it to cells that need it. So now let's start with our starting point. As I mentioned at the start, is eating and need for energy. And so let's consider this scenario of consuming a large meal. Now, a large meal can contain a multitude of macromolecules. These are natural polymers such as carbohydrates, proteins, and lipids that we need to survive. And most of the monomers of these polymers are converted to glucose because glucose is the be-all and end-all molecular fuel to make ATP. So this is why often when you consume foods, most of the monomers such as amino acids and fatty acids, and also other isomers of glucose such as fructose and galactose, etc., they're converted to glucose because this is the best molecular fuel that we use to make ATP. And we can actually use other we can use other monomers from other macromolecules such as fatty acids to make ATP, but it's a much more arduous and difficult process, and it requires much more reactions than just simple cellular respiration. But glucose can be put into cellular respiration immediately because it's the be-all and end-all molecular fuel. And only a certain amount of glucose, however, of the total intake that a person can consume in a large meal can be absorbed by the small intestine. And this small amount is immediately processed to make ATP for energy. In this case, however, with a very large meal, there is much more glucose floating around in the bloodstream than your cells need at the moment because it's a large meal, so it would contain a lot of glucose. So the excess glucose that is not needed immediately, in theory, it would remain in the bloodstream, and this can potentially cause quite a few very difficult problems. So therefore, a special hormone in the pancreas to regulate blood glucose levels is the notorious insulin. And this is where the bit about diabetes comes in. Insulin's job is carbohydrate uptake, and is to move glucose out of the bloodstream and into storage. And to do this, it triggers a shift from catabolic reaction, that's breaking down molecules to release glucose for energy, and instead it increases the rate of anabolism, which is linking glucose together in polymerization reactions to form glycogen. I will go into more detail about that in the next slide. But here's the mechanism that I'd like you to know. We don't actually need to know all of this. It's just excess detail. But the main important bit here is glucose transporters, these proteins here, and these are involved in glucose uptake into the cell, especially when insulin is used. Insulin is this known as a transcription factor, and it causes transcription genes, as you can see down here. We don't need to know this excess detail. Genes result in the protein being made. The protein in this case is this channel protein here known as the glucose transporter, and that allows for more uptake of glucose into cells, thereby reducing blood sugar levels to normal. And I'll send this flashcard in addition, so you can get a good picture of carbohydrate uptake here. And so the problem posed by diabetes, as I mentioned in the beginning, I'll mention it here, is well, sorry, I didn't mention the problem. I just mentioned diabetes as the name. The problem posed by diabetes is that your body either does not produce enough insulin, so there's not enough of there's not enough of glucose uptake into cells, or your body cells do not respond to insulin. They're not as receptive to insulin as they should be. But the result of these both types of diabetes, type 1 and 2, is the same, and that is glucose is not absorbed into cells fast enough, so therefore it accumulates in the bloodstream. As a result, blood sugar levels rise. And I'll mention two problems of this. There are a multitude of problems, but I'll just mention two and then we'll move on because of time. One big problem posed by this is the fact that glucose cannot enter your cells, so your body cannot produce ATP that it needs. So as a result, you may feel very, very tired. And also another problem that could be posed by very high blood sugar levels is the fact that sorry, lost my train of thought. Or is the fact that with the excess glucose in your bloodstream, your body cells may try to dilute the excess sugar to try and absorb them, and they do this by pulling fluid out of your cells. This actually happens spontaneously by osmosis, and because your cells, you can picture this in your head. This is it sounds as bad as it seems. Your body cells losing fluid to try and dilute the excess sugar in the bloodstream, it causes your cells to shrivel up, and this can leave you feeling dehydrated. So now let's look at some keywords that we'll need to know. We'll go into details about each one of these. Please note these down because these are very important. First, we have glycogenolysis. This is the process of breakdown of glycogen to release glucose into the bloodstream to raise blood sugar levels when needed. And this is controlled by the hormones the hormones glucagon and epinephrine, which is adrenaline. And adrenaline or epinephrine is involved in the fight or flight response. And essentially, glycogen is broken down and lots of glucose is released into the bloodstream to give you lots of ATP for energy to either fight or run away. That's the fight or flight response. And glucagon is the hormone released by the pancreas's alpha cells when your blood sugar levels are very low. And glycogenesis, the next keyword here, is excess glucose polymerized together to form glycogen for storage when you need it. This is controlled by insulin, as I mentioned with the flashcard process here. And the whole goal of all these processes here is to maintain homeostasis, that's constant internal environment. Because too much or too little of anything is bad. Even good things like water, for example, too much water can kill, and too little, as you know, can kill as well. Then we have gluconeogenesis. This involves glycerol, triglycerides, which are lipids, lactic acid, and amino acids. They're converted by the liver into glucose when needed. And these are controlled by the hormones cortisol and glucagon. Now, cortisol and adrenaline, they're both stress hormones. They release at times of stress. So they have very similar functions. That's why we can use cortisol and epinephrine interchangeably here when we're talking about these processes and the hormones that control them. Where glucagon as well, as I mentioned, it raises blood sugar levels, so it's also involved in this process. Because gluconeogenesis is very similar to glycogenolysis in that the end goal is the same for both of them, which is to raise blood sugar levels when required. And I highlighted gluconeogenesis in red because this is like an emergency process. This is the body's last attempt to raise blood sugar levels when when required, and this is an emergency process. This doesn't happen immediately. This happens as a last resort. That's why it's highlighted in red here. And next we have substrate level phosphorylation. This is transferring high energy phosphate group from an intermediate directly to ADP to form ATP. ATP, what it stands for is adenosine triphosphate, and ADP is diphosphate. So when you have ADP diphosphate, you add another phosphate group to that, that forms ATP, which is known as energy. And ATP is hydrolyzed, i.e. water is used to break it down, back to ADP and phosphate. So this is a reversible reaction. And this process of breakdown of ATP by hydrolysis, this is what releases energy for all your metabolic processes. So that's why ATP is very important, because our bodies hydrolyze it to release energy by breakage of one of the phosphate groups of ATP to form ADP. And we'll go into more detail about this in the next few slides, because substrate level phosphorylation is very important for cellular respiration. And then finally, for this slide, we have oxidative phosphorylation. This is removal of electrons from NAD and FADH2. And these are known as reduced NAD, because NAD is reduced, i.e. it gains electrons via the hydrogens, and FAD same as well, and pass them through the electron transport chain to oxygen. I will go into more detail about this. It's the last stage of aerobic respiration. And I'll just mention here, NAD and FAD, they are enzymes that control respiration, just as, for example, just as for example, there are enzymes, I mentioned amylase that digests carbohydrates. It regulates the breakdown of carbohydrates. It controls the reactions. Okay, so let's start with aerobic respiration. This is the very complex process that involves oxygen to produce ATP or energy. So here are some key points here. It occurs in the cytosol. The cytosol is the matrix or fluid of the cytoplasm of the cell, and it leads to the production of 2-pyruvate and 2-reduced NAD. And I highlighted these in blue. These are important, so take note of these, the numbers of these molecules. And NAD is a coenzyme, as I mentioned before. So this is the simple diagram of the overall process of glycolysis, the first stage of aerobic respiration, and that is glucose is converted into pyruvate. And if you ever get confused, look at the number of carbons. Glucose, 6 carbons altogether, 1, 2, 3, 4, 5, 6. Pyruvate has 3 carbons. So for every 1 glucose molecule that's put into glycolysis, we produce 2 pyruvate molecules. And also these points here are very important. ADP plus PI, which is a phosphate, equals 2 ATP. So here, substrate level phosphorylation occurs, as I mentioned in the previous slide, wherein ADP, adenosine diphosphate, is phosphorylated. Sorry, a phosphate group is added to it, and that forms ATP, 2 molecules of ATP. And we also have NAD, the coenzyme that controls, that regulates these reactions. NAD plus means it lacks, it's electron deficient, it lacks electrons. However, it's hydrogenated, meaning it gains hydrogen or electrons, and that forms NADH. And that's important for the next part of the reaction, for the next stage of aerobic respiration, sorry. And we have to go into more detail about glycolysis. This is not enough, but the detail is comprehensible. So here, we only need to know the detail about this first half of glycolysis here, 5 reactions. And these are some of the brief points. Glucose priming, this is what glycolysis is. It gets glucose ready to split and involves phosphorylating glucose, i.e. substrate level phosphorylation, adding a phosphate group, and also molecular rearrangement. That's converting between different isomers, because different isomers of glucose are actually made here. And I mentioned this, glucose and fructose are actually isomers. They have the same molecular formula, just different atomic arrangement. And so let's look at this enzyme here, hexokinase. It's the enzyme that triggers hydrolysis of ATP. So ATP is broken down by water into ADP and a phosphate. And as I mentioned before, this releases energy. This energy from hydrolysis for ATP is harnessed by glucose here and the enzyme uses this energy. The enzyme lowers the activation energy of this, and this is used to convert glucose, to phosphorylate glucose, to make glucose 6-phosphate. So the phosphate group from ATP is being taken off by hydrolysis here, and it's added to the 6th carbon on glucose. So this is a higher energy molecule than this because the phosphate group has been transferred. So this is substrate level phosphorylation. And this enzyme phosphoglucose isomerase is an enzyme that converts between isomers. So glucose 6-phosphate is converted to fructose 6-phosphate. Now this enzyme here, phosphofructokinase, is very, very important. That's why it's circled here. And this essentially regulates the rate of glycolysis. And phosphofructokinase is involved in substrate level phosphorylation again. It transfers a phosphate from one molecule to another when needed. And phosphorylation is the key of this whole process of glycolysis, and it also increases the rate of reaction. So this enzyme is phosphorylates glucose to increase rate of glycolysis when needed. Take note of that. And substrate level phosphorylation occurs, hydrolysis of ATP occurs again to form ADP and PI, which releases energy. This energy is required to convert fructose 6-phosphate to fructose 1,6-biphosphate. So 2 phosphate groups now. So fructose has been phosphorylated again. So this is an even higher energy molecule than this. And then finally, these are 2 compounds that we need to know. Glyceraldehyde 6-phosphate. Glyceraldehyde 3-phosphate, sorry. This is important because it's involved in, again, the hydrogenation of NAD plus to form NADH, and this coenzyme is very important for all of these reactions in aerobic respiration, and it's going to be used in the next cycle, so pay attention to that, as I mentioned in the previous slide. And glyceraldehyde 3-phosphate, yeah, it forms 2 NADH molecules that we need for the next step of aerobic respiration. So yeah, this is very important because it produces NAD. Moving on now. Here we have the next stage. The next stage, sorry, I forgot to label this as number 2. The next stage of aerobic respiration is called the link reaction. The link reaction links glycolysis to the next phase after this second one, to the third phase of the reaction of aerobic respiration, and that is the Krebs cycle. This occurs within the matrix of the mitochondria. So glucose in glycolysis is converted to pyruvate. Pyruvate moves into mitochondria, then undergoes the link reaction. So pyruvate can move into the mitochondria for this, but glucose cannot. So that's the importance of converting. That's the importance of glycolysis, and also the ATP and NAD produced. And this just came to mind. In glycolysis, 4 molecules of ATP are made. However, 2 are used up to convert between isomers in these multi-step reactions here. So overall, there's a net gain of 2 ATP molecules. That's why I'm very sorry I forgot to mention it here, but yeah, 2 ATP molecules altogether. So there's a net gain of 2 ATP molecules in glycolysis. Back to link reaction. This produces 2 reduced NAD or NADH and 2 acetyl-CoA. And pay no attention to that. It's highlighted in blue. And this will all be collated in the end as a summary. So the cytosol, which is which is where glycolysis occurs, pyruvate moves to the mitochondria to undergo the link reaction, and in the link reaction, decarboxylation occurs. So a carbon atom is removed from pyruvate. So 3 minus 1, which equals 2. So that's how many carbons that acetyl-CoA has. So this decarboxylation, it results in the release of carbon dioxide. And carbon dioxide is a waste product of aerobic respiration. So that's why we breathe out. We breathe out carbon dioxide because it's a waste product of aerobic respiration. And you've seen this in the overall equation for aerobic respiration, and I'm sure you know that. So then, decarboxylated, so it loses 1 carbon to form 2 carbons, and then hydrogen is added, and so NAD is hydrogenated again to form reduced NAD. It gains electrons, so it's a reduction, and this produces NADH, which we need again for the next part of the reaction. And then coenzyme A is added to the acetyl group that forms from decarboxylation of pyruvate, and that forms acetyl-CoA. So these 2 molecules, acetyl-CoA and reduced NAD, they are the take-home molecules from link reaction, and we need it for the Krebs cycle, which is the next stage. And these 2 points here are summarized real quick. 2 pyruvate molecules. So remember, as I mentioned, the glucose is 6 carbons. So for every 1 glucose molecule, we get 2 pyruvate molecules. And so for every 2 pyruvate molecules, we get 2 acetyl-CoA. So for 2 pyruvate molecules, we get 2 moles of carbon dioxide and 2 moles of reduced NAD. Okay, cellular respiration. Okay, sorry, the next process, the Krebs cycle, number 3. So this happens once for every pyruvate molecule, so twice for every glucose molecule, because 1 glucose forms 2 pyruvate. And this occurs within the matrix as with the link reaction. And this produces 1 ATP, 3 reduced NAD, and 1 reduced FAD. NAD and FAD are the coenzymes used to regulate all these multi-step reactions in aerobic respiration. So first we have acetyl-CoA, and the CoA is removed, and so the acetyl group is left. The acetyl group is 2 carbons. Remember the number of carbons, they're important. And this is combined with oxaloacetate, which is a 4 carbon compound, and that collectively forms 6 carbon compound called citrate. And then citrate undergoes a dehydrogenation, loses hydrogen. Those hydrogens are transferred to NAD plus to form NADH again. And also citrate loses 1 carbon, it's decarboxylated, and that forms another carbon dioxide, which is released as a waste product. And again, this is the 5 carbon compound, and the 5 carbon compound is decarboxylated again. That releases another mole of carbon dioxide, and it's also dehydrogenated, this 4 carbon compound again. Those hydrogens are transferred to NAD plus to form NADH, and we're going to use this in the final step of this process. All these NADH and FADH that we've accumulated before, we're going to use it in the final process. And also, it's decarboxylated, so that forms 4 carbon compound, which is yeah. And then this forms GDP and PI. And PI, as you know, substrate level phosphorylation occurs again, and this forms GTP, which is an energy carrying molecule similar to ATP. And also in this process, when this 4 carbon compound is converted to oxaloacetate, it's dehydrogenated. This compound 4 carbon loses hydrogen. Those are transferred to FAD to form reduced FAD or FADH2. And FADH2 and NADH, they're both electron carriers. The electrons are carried in the hydrogen atoms, and we're going to need this for the final process. Okay, so then NAD is reduced again to form NADH. Finally, oxaloacetate is formed, and that's used in the next link reaction. Then CoA, that's used in the next Krebs cycle, sorry, oxaloacetate. And then CoA goes back into the link reaction, is added to an acetyl group again to form acetyl-CoA. That comes back in the Krebs cycle, and the process goes over and over again. So overall, from two rounds of or 2 circuits of the Krebs cycle, for every 1 glucose molecule, we produce 1 ATP, 3 reduced NAD just from Krebs cycle alone, and 1 reduced FAD. So these coenzymes reduced NAD and FAD, all of them that we've accumulated so far from these 3 steps, those are glycolysis, link reaction, and Krebs cycle. We're going to use all of this in the final step, which is known as oxidative phosphorylation. So oxidative phosphorylation, this involves the movement of electrons through carriers in the electron transport chain, because electrons are moving along. That's what we call it, the electron transport chain. The electrons, they transfer energy due to their negative charge. So when an electron moves from one point to another, that transfers energy. And this energy is used to pump protons actively. Now, this proton movement is active. It requires active transport, it requires energy. So the energy that we use for this transport of protons, it comes from the movement of electrons, because that transfers energy. And this movement of protons creates an electrochemical gradient, which is a gradient formed by charged particles such as ions. This is known as chemiosmosis. That's the key word here. And this diagram, this is a simple diagram. We have to go into more detail as well, and that goes in the next slide. But this simple one summarizes it pretty well. This is the mitochondrial matrix, and we have low H+ concentration, not many here. and we have high concentration of H plus on the intermembrane space. So this process of proton pumping is active because it goes from low concentration of protons to high concentration. And where do these protons come from, though? In the beginning, the protons come from the reduced coenzymes, reduced FAD and NAD. So these are broken down. The hydrogen is removed, and those H2 molecules, hydrogen, they are split apart into protons and electrons. The electrons go into the carriers, and they move along through the carriers, and movement of electrons through them transfers energy or releases energy. This energy that the electrons transfer from movement within the carriers, that energy is harnessed by the mitochondria, and this energy is used to pump the protons actively. And these electron carriers are actually proteins. They're large proteins known as protein complexes, and we'll go into more detail about this now. So this is the more detailed process. Firstly, the complexes are numbered 1 to 5. 1, 2, 3, 4, and 5. And 5 is actually ATP synthase. ATP synthase is here. And actually, I should mention this before going on to more detail. Now, because the H plus ions move from low to high concentration, that forms an electrochemical gradient, as I mentioned before. And normally, nature doesn't favor gradients, so the protons have to move back into the matrix of the mitochondria to re-establish the balance, to get rid of the gradient. So then as they move back, they move through this protein complex known as complex 5, or also known as ATP synthase. And when the protons move from the intermembrane space to the matrix of the mitochondria using ATP synthase, this phosphorylates ADP to form ATP. And how this is the overall reaction of oxidative phosphorylation here. We have a half a mole of oxygen, so one oxygen atom, plus 2 protons from the matrix of mitochondria, so that come from the intermembrane space through here. And these are added 2 electrons within the carrier, and that forms H2O. So the reason we breathe oxygen is so it can serve as a final electron acceptor to produce water, and therefore produce maximum amount of ATP that we require for all our metabolic reactions. Oh, sorry. And oxidative phosphorylation produces the most ATP, and that's 32 in fact. Okay, so now the more detail. So complexes 1, 2, 3, and 4, these are the electron carriers. Now, I mentioned here that the electrons move within the carriers and they transfer energy. But what actually goes on? The electrons, they move through a series of collection of atoms within the carriers. Those collection of clusters of atoms are known as redox centers. And so the electrons move within the redox centers from one to another, like they started around like that. These occur within the complexes, and each time an electron moves from one redox center to another, so one point to another, that releases or transfers energy. And so collectively, however many redox centers there are, we can imagine that the electrons movement between the redox centers will produce a lot of energy overall. So all this energy from all these complexes is harnessed by the mitochondria, and then that mitochondria uses it for chemiosmosis. And these coenzymes here, these transfer electrons from one complex to another. So from complex 1 and 2, the electrons are recycled or used again. They are taken up by coenzyme Q, and they transfer the electrons from complex 1 and 2 to 3. Now this succinate fumarate is FADH2. So FADH2 is used in complex 2, and NADH is used in complex 1. So now complex 3, okay, the energy is harnessed, as I mentioned before, by movement of electrons within the carriers, and that uses the energy to pump protons again. And then the electrons that have moved within the electrons that have transferred energy, they move to cytochrome C. Cytochrome C is another coenzyme, and these cytochrome C transfers complex 3, the electrons from complex 3 to complex 4. And then finally, complex 4, the last of the protons from the matrix goes into the intermembrane space, is pumped actively via the energy liberated from the electrons. And finally, the electrons move out of complex 4, and then the protons move through ATP synthase from the intermembrane space to the matrix of the mitochondria. And so the electrons from here, complex 4, protons from complex 5, and oxygen, when you breathe it in, they're collectively added together for the overall reaction of oxidative phosphorylation. That's to produce water. So from half a mole of oxygen plus 2 protons plus 2 electrons, that forms 1 molecule of water. This whole process, when the protons move through ATP synthase, that triggers phosphorylation of ADP to form ATP. And I'll just mention this as a side point here. The reason why mitochondria is called the powerhouse of the cell is because the mitochondrial matrix is studded with thousands, if not millions, of these complexes that produce ATP collectively as a unit. So this is why the mitochondria is referred to as a giant cellular power plant or powerhouse, because it contains thousands, if not millions, of these essentially machines or complexes that produce ATP or energy. Now, the importance of this proton gradient or chemiosmosis is simple. That is, if there's no chemiosmosis or electrochemical gradient, there are no protons that move through ATP synthase, so therefore no ATP is produced. So if no ATP is produced, the cell will quickly become starved of energy and it will die. So that's the importance of this reaction. And a metabolic poison such as cyanide, what happens is that it serves as the final electron acceptor instead of oxygen. So instead of oxygen taking up those electrons from complex 4 here to make water to produce ATP overall, instead cyanide hijacks this electron acception, and what cyanide does is it reduces the amount of ATP that's made. So the cell becomes deficient in ATP, lower than normal, and it will die. Okay, these are all the blue highlighted words that I mentioned. These are all the molecules that are produced and enzymes from all the stages of aerobic respiration. And 38 overall total ATP molecules is theorized, but the real quantity is 32 overall. Finally, let's go into more detail about about these gluconeogenesis and glycogenolysis. So glycogenolysis, as I mentioned before, is breakdown of glycogen to release glucose for energy. And so we have glycogen here in glycogenolysis, and it involves multiple enzymes and also 2 steps. And these together, they form glucose to release glucose for energy, and blood glucose levels rise as a result of glycogenolysis. And gluconeogenesis, which is the emergency last resort process to raise blood sugar levels. It takes pyruvic acid or lactic acid and also amino acids and glycerol from lipids, and they are converted into glucose 6-phosphate, and then the phosphate is removed, and that forms glucose to raise blood sugar levels back to normal. Glycogenesis is this you're familiar with. This is part of glycolysis, but the key distinction here with glycogenesis is the fact that we use this enzyme here, glucose 1p umidyl transferase, and that converts UTP to this molecule PPI, and eventually this forms UDP glucose. UDP glucose is essential for glycogen formation. So UDP glucose, the glucose is removed to form UDP, and UDP releases energy required to polymerize glucose to form glycogen. Finally, anaerobic respiration. Anaerobic respiration is respiration without oxygen. So what actually happens is glycolysis still occurs, so glucose is still converted to pyruvate. However, without oxygen, we'll see that there are 2 byproducts that form with anaerobic respiration. From pyruvate, it ends up being converted into ethanol. This is one byproduct. And ethanol, as you know, is an alcohol and it can be very toxic to cells in very high concentrations. So that's one problem posed by anaerobic respiration, is toxicity. However, anaerobic respiration is also known to produce energy in short bursts. But why is that? The reason why is because NADH is dehydrogenated to form NAD plus and hydrogen ions, and NAD plus is the coenzyme that is used to make glycolysis to keep going. And as I mentioned before in the previous slide, let me just go back to it. NAD plus is needed to form for glycolysis, and it still produces ATP. So even with anaerobic respiration, we still get some ATP produced. So that's why anaerobic respiration is known to release energy in short bursts, but not the same amount as aerobic respiration. And also the other byproduct, sorry, is without oxygen, anaerobic respiration, we have NADH. Sorry, it forms lactate. And as you know, NAD plus is still produced, so again, glycolysis can still occur. The cell can still make ATP, but just not as much as aerobic respiration. And lactate is a precursor to lactic acid. Lactic acid is, as you know, is an acid. It dissociates, readily releasing lactate and hydrogen ions. And so because the protons or hydrogen ions are released, that results in cell pH decreasing. And as you know, the enzymes, in this case, coenzymes NAD and FAD, will denature, and other enzymes as well that are involved in all these reactions. And so the reactions won't be regulated or catalyzed, and therefore they will just stop altogether. So those are the problems caused by anaerobic respiration. That's the end. I hope you learned a lot from this. If you did, please let me know. And if you have any questions or still some gray spots, please let me know, and I'll try and help as best as I can. Thank you for watching. Take care.